Okay, good morning, everybody, and welcome to the 2023 edition of the Royal Tyrrell Museum Speaker Series. My name is Caleb Brown. I'm one of the curators here at the Royal Tyrrell Museum, and I'll be your host today. Before we begin, I would like to acknowledge that the speaker series is made possible through the generous support of the Royal Tyrrell Museum Cooperating Society. Today, the Royal Tyrrell Museum and its Cooperating Society are proud to present Dr. Yara Haridy. Yara is currently a postdoctoral researcher at the University of Chicago in the Shubin Lab, studying the early evolution of mineralized tissues in vertebrates. Yara got her paleontological start by volunteering and then progressing to do her master's at the University of Toronto, Mississauga, where she studied the histology of tooth replacement and development in early reptiles. Yara completed her PhD in Berlin, Germany at the Natural History Museum there. Her thesis focused on the evolution of osteocytes and bone cellularity in the fossil record. She pioneered the use of FIB-SEM, which she'll talk about today, tomography, for nano-level imaging of fossil cells. She's currently searching for the earliest mineralizing vertebrates in the fossil record to better understand the origin of our skeletal biology. When not cutting up fossils, Yara is committing her time to DEI efforts in paleontology and science communication. Today, Yara will be presenting her research on the origins of fossil bone titled Deep Time Beginnings of Bone Biology. So without further delay, I will hand the mic over to Yara.
Perfect. Thank you so much, Caleb, for the wonderful introduction. So, hi everyone, I'm Yara Haridy. I'm a postdoctoral researcher at University of Chicago, as Caleb has already told everybody. And I study bones and everything mineralized, basically, particularly in deep time. So that means fossils. But first, a little bit about myself. So I'm an avid science communicator. I truly believe we understand the deep time and Earth history. The more we understand it, the more we can make sense of our own world today. I do a lot of science communication, and these are some examples of what I do. So on here, what I'm holding is a little mock-up of a puppet that we are building for the education of the evolution of flight and feathers. So this should be an accurate little velociraptor. It's still in the works. Additionally, I do a lot of work with museum-based specimen outreach. And what that basically means is I just take a fun specimen, walk around a museum, and talk to people about it. This example here is called cactus antlers. They're a pathology caused by a virus, and it causes excessive branching in antlers of just normal red deer and mule deer. So this was a fun one to talk about because nobody has any idea what it is, and you get to talk about how bones grow and all the mutations that can happen. So in general, I don't really discriminate when it comes to what I study. I work on the very big and the very small. Here is me holding a very small little humerus with a pathology on it. So you can see a little growth right there, very tiny. And then a really big humerus of a sauropod that we dug up a couple of years ago in Utah, still in publication. Very exciting. So I focus on the hard parts of our biology. I don't mean difficult, but I mean physically stiff, non-squishy parts. This basically means I wake up every morning and I ask, why are we not goop? I always try to pitch this as my talk title, but people tend to want something more scientific or something a little bit more informative. So, why are we not goop? Many animals are goop. Most non-plant life on Earth is some form of goop. So, what I mean by this is, here you can see an amoeba. Amoebas are pretty categorically goop. Nobody argues with me on that one. Jellyfish in their adult form are mostly water, and then a jelly-like substance called mesoglia, which seems like pretty much the definition of goop. So no hard parts in sight. So I ask again, why are we not goop? Why do we have any hard parts at all? And the simple answer is really because of a process called biomineralization. And all that means is it's a process where organisms use minerals to generate hard structures like teeth, skeletons, and shells. Some of the most ancient forms of life have these skeletons. So these are sponges. Sponges make their skeletons out of silica, so the same thing that's in sand and glass. Shell-making animals make their shells out of calcium carbonate. They make their shells one little section at a time, and they keep adding little sections and not actually growing the old sections. And then, of course, the vast majority of animals, being invertebrates, also have a bit of mineralization. So mostly chitin, which is a type of fancy sugar, but also they include calcium carbonate within their exoskeletons. Now, what these tissues here have in common is that they're dead on maturity. And what that really means is that once it's mineralized, once it's excreted, and once it's deposited, they're not being remodeled, they're not changing with the animal, they're not growing with the animal. That's why crabs have to molt. That's why most insects have to molt, and that's why shelled animals grow a new chamber every time they need to grow bigger. So the hard parts don't grow with the soft parts, if you want to think of it that way. Kind of like our own hair, which is dead on maturity. So once it actually exits the scalp, it doesn't cause you pain, it can't change. It's basically just dead material. And this is where our skeletons are incredibly different from the vast majority of all other skeletons. So we're going to focus on vertebrate skeletons today, and particularly vertebrate bone, which is a very special tissue, and I hope you'll be convinced of that by the end of this. So what makes our skeleton different? One, it goes through a process called remodeling. This is used for both growth and adaptation. So as we grow, our bones grow as well. But as you can tell, we don't just keep accumulating bone as we grow. Some of it is taken away and some new bone is put in place. And this continuous reworking makes sure that our skeleton is perfectly adapted for our life as adults, as large animals. Additionally, if you take up running or you start weightlifting, your skeleton will also adapt to that. It will start to add calcification. It will start to adapt to your new mode of life. Our skeletons also regenerate, they heal. So if you break your bone, not only does the skeleton mend that break, but if there's even chunks missing, it will be remade. So this type of regeneration is not present in, say, clamshells or snail shells. They can close a hole, but it will never look exactly the same way it did. It will never function exactly the same way it did. And that's something our bones can do. And finally, and we'll focus on this a whole lot more, it's a living tissue. So what that means is it's fully cellular. There's cells inside, around, underneath, everywhere around our bone, and it's continuously living. This is why breaking a bone hurts. There's innervation, there's blood. It is a completely integrated system that's not really dead on maturity. It's in a living tissue. And most importantly, our skeletons are made to move. Our skeletons today are mostly internal. They support our muscles, they protect our organs, they're highly adaptable, and I, for one, I'm super thankful that I don't have to shed my skeleton regularly, because that sounds really uncomfortable. So vertebrate skeletons differ significantly from invertebrate skeletons in some of the ways that I just mentioned. And the reason that we call it a living lattice, we'll focus on first. So since vertebrate bone is living rather than dead, we can study it at several levels: the cell level, the tissue level, and the organ level. And all three levels can also be studied at an evolutionary scale. But since this talk is my talk and everyone has a favorite child, I'm going to start with the cell component since that's kind of my favorite. But first, there are many questions about the evolution of our skeleton that have yet to be answered. Like, what is the first bony animal? What is, or how did bone come to form? Where did it come from? Did it actually evolve from a different type of tissue and then a few genes changed and then now we have bone? Is it completely novel? Where did the cells come from? And could it always heal, or did that come later in its evolution? There are so many questions that seem very basic to bone biology that we still don't know and can almost exclusively be answered with the fossil record. So I look at fossils. Particularly, I look at bone cells, but I also look at pathologies, and they come together to actually inform bone evolution together. I promise it all ties in together at the end. So let's start with bone cells. We're going to go just super briefly over cell types. Let's talk about basic bone biology, which is informed almost entirely by modern animals. So everything that we see in our textbooks is almost fully informed by humans or modern animals that can be easily experimented on. So as you can see, this can probably cause a lot of bias when you start to look back into the fossil record. So the first cell is the osteoblast. This one's kind of easy to remember because it has the letter B in it. So osteoblast, bone building, osteoid producing, and collagen producing. They are the builders of bone. Osteocytes is what an osteoblast becomes. So a mature osteoblast, they're super interconnected, they're long-lived, they maintain bone, and they're all over our bones. We have, I think they are the second most interconnected cells in our body, only second to neurons. So that tells you something. And actually the way that they make their little processes, they express similar genes to neurons. So that's how interconnected osteocytes are. And then osteoclasts. These are these bone destroyers or bone dissolvers. They're not related to the other two cell types. They're their own type of cell. They're closer to your immune cells. They produce like an acidic substance that literally eats away bone, and they're incredibly important for our growth and bone remodeling. But we're going to focus mostly on osteocytes for a moment, because these cells are very special to paleontologists, like me. In paleontology, we have indirect evidence for all three cell types that we just went through, but really only direct evidence for osteocytes. And all that means is that we know that osteoblasts existed. We know osteoclasts existed because bone was produced and bone was resorbed. But the cell itself does not preserve in any way. And osteocytes don't preserve in themselves, but they preserve in their caves, this thing called the lacunae. And I'll show you. But first, osteocytes were discovered through paleohistology. I am a paleohistologist. I cut up fossils. We grind them really thin so that you can look through them, shine light through them with a microscope. Basically, the fossil is embedded in plastic, cut and ground super thin, and then we image them. And you can image something like this. This is under polarized light. That's why you get all these funky colors, which I like. But also what we can see is osteocyte lacunae. So these little black dots here that have all the fine lines in between them are the osteocyte lacunae, the little caves that the cell itself used to live in. The cell itself, of course, is not preserved. These are very, very old bones, and soft tissue does not readily preserve. But the cave itself is like a perfect little cast of it. And in this case, it's filled by another mineral, so you get this beautiful contrast. We also get nice preservation of primary osteons, where blood vessels come in and deliver nutrients to these cells, deliver nutrients to the bone, and also remodel the bone itself. So the lacunae of osteocytes is so perfectly to shape of the cell that paleontologists can estimate all kinds of things. We can actually know so much more about extinct animals than we ever used to be able to. So you can estimate genome size, for example, because the bigger the genome, the bigger the cell. So those two tend to correlate, and this has been ground truthed in modern animals. You can also estimate red blood cell size in a combination of measuring the primary osteon, where the blood vessels would go, and the osteocytes. Together, there's an equation that gives you red blood cell size. And red blood cell size is correlated with oxygen efficiency. And that also tells you about how the animal lived. Could it run really fast? Could it, how metabolism, how high of a metabolism it has. And this relates, of course, to the last point, which is this relates to metabolic rate. And metabolic rate can tell us a little bit about ecology and lifestyle of extinct animals. So why can osteocytes tell us all these? Or why can osteocyte lacunae tell us all this? Osteocytes are predictors of many things in physiology, particularly bone physiology, because they have very important roles. And again, these roles are basically dictated by the medical literature. So bone remodeling, we've gone through a little bit, partially growth, partially just changes in adaptation. Osteocytes live in bone. They send out signals to osteoclasts and osteoblasts to come build and come destroy. where it's appropriate, also for micro cracks and just general growth. They play a strong role in mineral metabolism. They live in these little caves, they live in their lacunae, and they can erode the lacunae and actually release calcium and phosphate into the blood system. And what that does is it buffers our blood, so your blood doesn't have to go wildly too acidic or too basic, but it also allows continued physiological function, even though, say, calcium is scarce right now, or phosphate is scarce right now. So that gives animals a really good advantage. If you don't have calcium right now and you can reuse your bones, you don't have phosphate right now and you can reuse your bones, that's what osteocytes do for you. And finally, mechanosensation. So stress, they can basically sense stresses on your bone and either initiate remodeling, initiate metabolism, or any other healing things. So they seem pretty important. Seems like osteocytes are a good deal in bone. Therefore, it's easy to be led that osteocytes are indispensable to bone physiology. Basically, all medical research is screaming that osteocytes are so important to bone health and basic function. It's a really fun party trick, for me at least, to tell medical researchers that actually the vast majority of vertebrates ever don't have osteocytes. They have something called anosteocytic bone. And so that means most non-jawed vertebrates, so everything really early, our earliest ancestors, and then secondarily lost in the most speciose group that's alive today, modern teleos, so like tuniforms and the vast majority of fishes. 50% of modern vertebrates fall within this group, and they have no osteocytes. So what is anosteocytic bone? What does that even mean? It's bone that contains both other cell types, but no osteocytes that are embedded. So here what you can see is a cross-section of a swordfish bill. Each one of these holes would have had a blood vessel. All these lines show the remodeling, so there is remodeling happening in the bone. But as you can see, there's no little dots all over, which is very different than the horse here. So we have a section of a horse, and you can see, again, lots of osteons where the blood vessels would be, many little dots because there's many little cells. So osteocytes, no osteocytes, but they both still function. In fact, an osteocytic bone seems to have mechanosensation just fine. This has been shown experimentally. They can clearly remodel. We can even see it here in this picture. But what they're going to always distinctly lack is mineral metabolism, at least at an efficient enough level, because that's a lot of surface area that these osteocytes have internally. And if each one of these can slightly eat away at the surface and deposit calcium or phosphate, that's a lot of surface area that these animals are losing out on. Modern fish, maybe it doesn't matter. Calcium can be obtained from the ocean, from the water. They can absorb it through their gills. So maybe it doesn't matter. But then that brings up the question of why evolve osteocytes in the first place? It's not like they're a neutral cell. They are very long-lived. Some of the most long-lived cells in our body. They're metabolically expensive cells. They add holes to your bone. So if you can do all the bone things without them, why evolve them in the first place? Could it be because of mineral metabolism? So this was one of the questions I really wanted to answer during my PhD. What if mineral metabolism is the driver for cellularity in bone? Now, this isn't a simple question, but it's not impossible. I started by sampling two main groups. The first, osteostracans here, which are basically jawless, mud-slurping fish from all the way from the Silurian to, I believe they make it a little bit into the Devonian. And they're the first to evolve osteocytes. And then I also sampled some antiarchs, these stem group jawed fish, so they kind of have a jaw. And they're the first to do secondary osteons, which are supposed to be a way of remodeling your bone, and it's based on, again, osteocyte orchestration. These are two good samples for just seeing what is up in early bone evolution. This is what the animals kind of look like. I apologize for the general lack of fossil pictures in this talk, but bear with me. So it kind of looks like a tadpole, no fins, very simple, little early fish. And then a slightly more advanced fish, you can see pectorals. There's kind of a beginnings of a jaw, eyes are on top. They're still all bottom feeders. Started with histology because I'm a histologist. Cutting things is usually how I start to discover what they're doing. And while paleohistology has been in use for a long time for understanding ancient tissues, there's kind of a major problem. Histo is easy, but this gives us 2D data for what is inherently a 3D structure, right? Osteocytes, the best part of them is that they're so complex in 3D. They have all these little processes that intricately branch and talk to each other in 3D space through mineralized bone. So I quickly found out that my favorite method was probably not going to work for me here. And I started CT imaging or CT scanning. We use synchrotron, which is just a really, really strong X-ray, basically. And this is a block of bone from Bothriolepis, that antiarch, the guy with fins. We were able to get really beautiful images and segment out canals and segment out even cells. However, we could see the cells. We can even map morphology to them. So the colors represent how long the cell is or how round it is. The more round, the more blue. The more long, the warmer color. But of course, the resolution that you get from scans is so tied into how dense your fossil is. And I'm working with very, very old bone, which means it's unfortunately very dense material. So this was the ultimate resolution we could get for the cells. As you can see, we can't resolve cell processes, right? So in some ways, histology is still better than this 3D rendering of what are essentially pretty colorful blobs. So what now? At this point, I was getting ready to kind of give up. Maybe my PhD question wasn't going to work out. Maybe the technology just wasn't there yet. And this is when I got to meet some colleagues who were also scanning, and they work on lithium batteries. They work on the erosion and deterioration of lithium batteries, and they showed me a poster that looked somewhat like this. And of course, you know, a hammer sees nails everywhere. So I was like, oh my God, these look like osteocytes. But they weren't. They were pores within the lithium battery, and they're seeing how they degrade over time. And I asked about, what is this method? How are you getting such amazing resolution at such tiny scale? And they basically told me that they use something called FIB-SEM. And FIB-SEM is focused ion beam scanning electron microscopy. And all it is a fancy schmancy machine that has two beams aimed at a surface. This is the FIB beam, and the blue one, it'll play again. And the blue one here is the scanning electron microscope, which all of us have used at some point. It's quite common in paleontology. So what this does is basically evaporate parts of the material. So the FIB is a focused ion beam, it's gallium, and it will actually evaporate and basically shave off the surface, and the other one will image. And they'll do that serially through the specimen. So what you can see here is we basically dug a hole. So this is the surface of the fossil bone, and there's a little pit in here. And inside that pit, what we can see is what looks like an osteocyte lacunae. And so we basically image that surface and then etch it so it disappears, and you image it again, and so on and so forth. And what you get here, well, first we actually used machine learning to like clean up the data. But then after that, what you got was a stack of images. And from any stack of images, you can create a 3D model. And basically, this is what was the very first 3D model of the earliest bone cells that are known to paleontology. And this was very exciting because at this point, we can actually, you know, see where they're talking to each other. We can start to calculate volumes. We can start to calculate how long of a distance they have from one another, which is pretty amazing. But the better find was this. So I'm going to do a little bit of CSI here and enhance. And so what you can see here is basically a halo of area that's demineralized, an area that has less minerals. And so we segmented out these areas, and we got basically this, a model that shows that the areas are not randomly placed, but they're actually perfectly placed around the cells and their junctions. And this is pretty strong evidence for a physiological process. And so just to prove it a little bit further, so this is the 400 million year old jawless fish, and this is a modern lactating mouse. So PTH is parathyroid hormone, and that's released when there's lactation. And so you can see a zone of demineralization here as well. So basically what we found was mineral metabolism in 400 million year old jawless fish, which is really, really small physiology. So just to put it in context, fine beach sand is about 90 micrometers, 70 is about a human hair, red blood cell is about 8 in diameter, and our biggest cell that we were able to resolve was just under 9. So very, very small physiology and really ancient. And this was evidence for cellular bone evolution. One of the theories that was always thrown around was maybe it's because of mineral metabolism. Maybe having this option, so the creature here that started to have osteocytes had a quick advantage of above some of the other mud slurping fish that lived alongside it, because it could maybe migrate. It could maybe resist starvation a bit more or continue physiological processes further because it continued to have osteocytes. It could do those things. So all this means is the earliest osteocytes seem to be able to do what all later osteocytes can. This means that it's an ancient trait appearing early in bone evolution. A lot of times we think of evolution as a stepwise acquisition of things. So maybe osteocytes should have first, evolved this and then that. But it seems like all of them appear around the same time. It's like a whole tool set that appears with osteocytes. So it seems that perhaps this is why osteocytes evolved in the first place. Maybe it's mineral metabolism. But of course, there's so much more work to be done. So let's shift gears a bit. I promised you that it wasn't just bone cells and little specks on a slide that we were going to look at. But I also look at something pretty different, pathology, particularly paleopathology in the fossil record. And this is because while we are able to see osteocytes, we are not able to see osteoblasts and osteoclasts or any of the other cells that are involved in bone formation, bone metabolism, bone remodeling and destruction, right? So as I said earlier, there's indirect evidence for these, but no direct evidence, as in we cannot see them and measure them. So this is where pathology can actually be super useful because it shows us what happens when some of these cells are not doing their jobs perfectly. And I'll explain that further. But first, what is paleopathology? It's literally the study of ancient diseases in deep time. It helps us understand origins and evolutions of disease, sometimes prevalence, especially in anthropology. People look through this to see if certain diseases have become more prevalent. So early plagues on humanity. So this is an example of an early osteosarcoma in a hominid. And then my baby here, which is this other osteosarcoma in an early stem turtle. And why study paleopathology, right? They're just bumps and lumps on what basically are rock, right? It's just fossils. But the problem is, without paleopathology, we have a black box phenomenon, especially about other cell types, not particularly osteocytes, but when you have a fully formed, beautifully preserved fossil, you have to assume that all biological processes are running well, and they're running in the same way that modern biology is running. We have to kind of assume that because we have nothing to show the contrary, but we don't really know. However, when we have some pathologies, they indicate that something isn't working right. Certain pathologies are indicators of cell abnormalities. This shows us or allows us to understand more about bone physiology in a certain point in Earth's history. If you're still a little confused, don't worry, so am I, but we'll get through it. So, like there are many types of cells, there are many types of pathologies. Of course, there can be mixes of multiple types, and this is a grand overgeneralization of the types of pathologies that can be found. Excuse me. So these general bins just kind of help us for the sake of this talk, understand why we're looking for certain types of pathologies. So in this case, traumatic pathology is what it sounds like. There was some kind of trauma that happened. In this case, it's a break, and then there's a bony callus that keeps stability of the bone while it heals and regenerates internally. And then eventually these parts are taken down and the bone looks pretty much good as new, sometimes even stronger. Developmental pathologies are incredibly informative because they can contain certain abnormalities that can indicate a problem in certain genes. So this can be indirect proof that those genes even exist. So certain like segmentation genes, like how our vertebrae work and which somites become which vertebrae. There's a lot of vertebral abnormalities in the fossil record, and this shows us, okay, these animals developed this way because this is where it goes wrong. This is where developmental pathology can be really helpful. Additive pathologies are basically an indicator of upregulated osteoblasts, so cells that make too much bone, or downregulated osteoclasts. There's not enough of the cells that are supposed to be shaving down bone. Or even a disturbance in the periosteum, the covering on bone that keeps bone alive. So additive pathologies can be indicators for several of those things. Erosive pathologies, where bone is taken away or there's too little bone in certain areas, can be an indication of downregulated osteoblasts, too little builders, or upregulated osteoclasts, too many destroyers, or a mix of both, as well as other infections and things that like cysts in bone will also cause erosive pathologies. And then finally, the last category is neoplasms. Neoplasms are one type of tissue changing into another. Often in humans, this can cause cancer, but it's not always cancer. So in this case, it's like bone that starts to change into cartilage and makes a blob of cartilage. Sometimes absolutely harmless, other times can be a problem. But this can be an indicator that gene regulatory networks are not working properly, or there's certain tumor suppressor genes that are not present. And of course, once I start talking about paleopathologies and all these categories, people are like, what are you talking about? How can you even know if your fossil is sick? How can you diagnose a rock, right? Which are totally valid questions. So how do you know if your fossil is sick? They won't cough, they won't take a COVID test, they won't go to the doctor, but sometimes all you need is a really close look. And I encourage everybody who's listening, next time you're at a museum, look at the big, especially the theropod mounts, even if it's just a cast, take a really close look, particularly at ribs, particularly at limbs. Sometimes all you need is a good set of eyes and just paying attention a little bit. So this one's very obvious on this allosaur. Allosaurs are often covered in pathologies. I'm assuming they were just big roughhousers. But this one's obvious. It's a rib that has a fracture callus. So it's a rib that clearly was not immobilized, because you can't really immobilize ribs, particularly in the Cretaceous. So, and here you can see a fracture callus, basically an overgrowth of bone that's supposed to stabilize the two ends. So this one doesn't look innocuous. It doesn't look like it could be anything else, but it's very likely a traumatic pathology, which is an indicator of either behavior or something that's happened to the animal at a certain time. Sometimes things can be a little bit more complicated. So here's an example of a plesiosaur. I was at a museum and I was looking at mounts really close, and I noticed that this tail just didn't look right. And as you can see, these two bones, there are all these vertebrae, they're all tail vertebrae, and these two are fused. Now, there's several causes to fusion, and we still don't know for this particular one. It's being scanned, but it can be arthritis, it can be trauma, and there's many other diagnoses that can cause vertebral fusion. And we'll get back to one of them at the end. So if it doesn't seem obvious externally, you need other methods. So as a histologist, again, I'm going to go for the saw, right? Sometimes the saw will help you, with microscopy, of course, figure out bone interactions on a cellular level or a tissue level, and then you can tell what kind of disease it was, because we have a really good library of modern diseases and how they interact at the tissue level, and then you can compare them. The other option when curators don't let me cut their bones is CT scanning. So sometimes that works just fine, particularly for things this young. So this is an ice age frog. Let me play that one more time. And what it shows here is a fracture callus. You can see the rest of the bone still exists, but it's been displaced. The two are no longer meeting, and there's a giant fracture callus holding both of them together. So simple scan, simple diagnosis. So other times the diagnosis can be much more complex, and a scan doesn't really give you a quick answer. So this is another case study we can do really quick. Popocales rosinae is a Triassic stem turtle from 240 million years ago. And I was given this little femur with this growth on it and basically told, hey, Yara, figure it out. I'm like, okay, let's try this. So we scanned it. And sometimes part of diagnosis is relying heavily on something called differential diagnosis, basically making a list of what it's not and why it's not those things. So we could tell there's no evidence of resorption. It's kind of hard to see here, but the bone is still intact underneath the growth. No evidence of a broken surface or any healing. Again, the bone underneath the growth is perfectly intact. No evidence of infection because there's no erosion and no bite marks. So all we have is this additive pathology, this thing that grew on top. And I'm not going to bore you with all the details, but long story short, it's a very special type of cancer. It's a cancer that lifts the periosteum, and we only know this because of really good medical literature. So it's cancer. And it's not the only fossil cancer either. In fact, cancer has been found in dinosaurs. This specimen is actually from the Tyrrell, and it was worked on by a team in Toronto and McMaster, and it shows a very pervasive osteosarcoma, a spreading bone cancer that started at the joint and went through the medullary cavity. So we have multiple fossils with bone cancer. What does that mean? People always ask me, well, so how old is cancer? Everyone thinks it's a modern phenomenon because of our modern lifestyle, because of the pollutants. And in reality, while that might be exacerbating certain things, cancer is actually incredibly ancient. So it's a little bit complicated, and it depends on what kind of evidence you're willing to accept. So there's some examples that are pretty old, but it actually gets much older. There's a really amazing paper that we're going to talk about here, and it reviews cancer-like cells across the tree of life. And basically, there's some form of cancer or uncontrolled cell growth all the way down the tree, all the way down to multicellularity. And what this paper says is something really amazing, quite elegant. And cancer is the rule breaker. It is breaking rules of 5 components of multicellularity. To become a multicellular organism, you have to agree to certain rules. And what that means is no uncontrolled proliferation. You can't make just your cell type, don't make too much of it. Resource monopolization. Your cells cannot take more than everyone else's cells. Dysregulated differentiation. Oh, you're becoming all kinds of cells. That's not what we agreed on when we all signed up for multicellularity. Environmental degradation. You don't want to be degrading the tissue that you and other cells are living in. And inappropriate cell survival. If one cell is living for way too long and monopolizing everything. So any breaking of any of these rules can actually cause basically cancer. And these are the rules of multicellularity. So that means cancer is deeply tied to the evolution of multicellularity. So now what? If it's so ancient, then shouldn't all animals get cancer equally? Well, that doesn't seem to be the case, right? It actually, theoretically, you should have more cancer in animals that are larger and that live longer. Those animals have more cells and they live longer so they can accumulate more mutations. That makes sense. By that logic, large old animals should have the highest rate of cancer. But that's not really what we see, is it? In fact, what we see is kind of a balancing act. So for example, elephants have a really low rate of cancer. And take this next part with a grain of salt. But we don't have a whole lot of cancer in dinosaurs. Of course, that could be selection bias or preservation bias, but we still don't have them. And this is called Peto's paradox. And this leads people to actually research cancer in different animals. Why is this happening? Why do the animals that have more cells and live longer not get more cancer? And we'll go quickly through this part just because it's not my own research. But basically, long story short, animals are not static piles of cells. They adapt. They try to fight their own inherent biology, and they want to live longer without cancer. So things like elephants have 20 copies of tumor suppressor genes. So they're pretty set. Naked mole rats live in low oxygen environments, and they're pretty much immune to cancer, even when cancer cells are introduced to them, and they're incredibly long-lived. And the way they do this, apparently, is by having their cells hate being crowded. They have a mutation that allows their cells to not want to be so crowded, and therefore that in itself suppresses tumors. Rodents naturally have a lot of cancer, except somehow the biggest rodent. The biggest rodent has the lowest amount of cancer. And that's because of an expanded family of immune-related genes that made their immune system hypervigilant for cancer cells. So they have an adaptation against it. And the same thing with many species of bats. They live super long and have very stressful lives, but they have extra copies of certain tumor suppressor genes, as well as things that stop their telomeres from degrading, these things that are at the end of your DNA. So basically, they have adaptations against mutations. And so this explains some of Peto's paradox. Some animals that might have a higher rate of cancer actually have strong adaptations against it. So does that mean cancer is rare in dinosaurs because they had a lot of anti-tumor genes? We don't know. And realistically, we might not know. It might be selection bias, but only more research will tell. So if you're ever looking at a bumpy, lumpy fossil, it could mean a whole lot more than it's just a sick animal. It could actually help us figure out if dinosaurs and other animals, other ancient extinct animals, had cancer-fighting properties. Now, let's get back to the bigger picture of cellularity and what fossils can tell us, particularly pathologies. So again, I can't understand the physiology of a perfect fossil. If an animal is healthy, we assume it's working fine, and it tells us very little about what's happening inside. So we're going to do one last case study, and this is actually my own work. So this spinning vertebrae is a normal vertebrae, so not a pathological one, of a Permian reptile called a Baranopid. And it's a fun-looking little vertebrae. It's fine. This one looks a lot less happy. Here's two Baranopid vertebrae further down in the tail, and they're fused together. That's fine. Life happens. A fusion is not the end of the world. But externally, the fossil itself looks quite smooth. There's no indicators for a break, anything like that. So of course we scan it. And this is where you can see the internal structure. And this was a really strange puzzle for me for a long time. And here's why. In certain areas inside the vertebrae, you see these big scalloped holes. That's not normal. And here, where they fuse, there's just an overbuilding of bone. In fact, there's an overbuilding of bone at the bottom and an overscalloping of bone internally. And it seems like this has happened in two stages. So there was a lot of resorption and then a lot of building. And I was very confused for a long time staring at this until I read about some metabolic bone diseases. The way metabolism in the bone happens, the way resorption happens, can become dysregulated. In this case, it's called Paget's disease. It's actually a very confusing disease that happens in humans, sometimes related to viruses. We're not actually sure why it happens still. But this is what it does. It dysregulates the cells, and then all of a sudden they're overbuilding, and then all of a sudden they're overabsorbing. And so what we have here is a 290 million-year-old Permian critter with a metabolic bone disease. It's a little late to help him, but it tells us a lot. And what it tells us is this. Now that we have an erosive pathology and an additive pathology in one specimen, it tells us that there's likely an upregulation of osteoclasts and a downregulation of osteoblasts. And better yet, that they're related. They're talking to each other, and that communication in between them can actually be broken. This indirectly shows that communication between osteoclasts and osteoblasts existed in the early Permian. And of course it existed in the early Permian. They were growing and resorbing normally. But this type of experiment or this type of thinking and this framework of looking at pathology can be applied to much, much earlier animals. And that is how we will start to understand how mechanisms break. Basically, when a mechanism breaks, we can actually understand how it works. It's like when your toy breaks open, you can start to look at its mechanics inside. And this is why I look at pathology. The long story short, each one of these pathologies tells us so much more about the animal and its physiology, and more importantly, the cells that don't preserve. It tells us on a biological level how it heals, how it interacts with viruses and its environment, how it interacts with trauma. So there's so much more information wrapped up in these pathologies. And what we really need is a library of these deep time pathologies. So I always encourage other paleontologists, if they have a quirky bone, to please describe it. Even if it's just the briefest of descriptions, these will allow us to look at long time trends of bone evolution and bone reaction. So I hope I've convinced you that pathology, along with the knowledge of bone cells and bone physiology, can actually inform bone evolution, and how fossils are so intricate to this and to our understanding of why we're not just goop. Why do we have hard parts and what they do for us in the first place? So I'd love to take any of your questions related to this talk, bones, or paleontology as a whole. But thank you for listening. And don't forget to thank your skeleton for all the support and all the work it does for you on a daily basis. Thank you so much.
Okay, thank you very much, Yara, for that great talk about bone biology and some of the cool research that you're doing. We're going to have time for some questions. So if you have questions in the audience, just use the Q&A button at the bottom. While those questions start loading up, I will remind everybody that next week, April 13th, we will have Dr. Taryn Grant from the University of Sao Paulo in Brazil. He'll be talking about chemical defense in poison frogs. We see there's a couple of questions coming in. The first one is, why are most biomineralized tissues comprised of either silica, calcite, chitin, or cellulose? What intrinsic properties of these materials are ideal for biomineralization? And given that biomineralization has evolved with the same material convergently many times. That's a very good question.
Yeah, that's a great question. So basically the question is why these materials versus, for example, our own skeletons, which are calcium phosphate, right? So first of all, I'll just point out that chitin and cellulose are technically not always biomineralized. Sometimes they are, sometimes they are and sometimes they aren't. It just depends if they're actually including a mineral aside from just the sugars that they're made of. But that being said, why calcite and why calcium carbonate, for example? Why is that so common? And the reality is probably because, at least in calcium carbonate, the marine environment allows for so much of that material to be readily available that then can be sequestered by the animal and then redeposited, at least for calcium carbonate, so like shells and stuff like that. It's so intricate, in fact, that's why, for example, our global warming is becoming a problem. Acidic environments don't allow for certain shell formations. So it's probably incredibly tied with the environment, what is available and what can be utilized. And it's been shown in the past, I believe in the Ordovician, maybe later, don't hold me to that, but in a previous climate change event where the ocean also acidified, animals actually switched from using calcium carbonate to calcium aragonite, I believe. So it's not like it's super stable, but it seems to be probably a balancing act between what's like metabolically costly. So some minerals are more metabolically costly to work with, and what's available in the environment. And then, of course, phylogeny. If you're related to an animal that is making its shells out of calcium carbonate, you're way more likely to also make it out of, so success, right? Success in phylogeny will kind of bottleneck certain types of mineralization. Like, why don't we see so many more types, right? But that's probably why.
Hey, great answer. Next question is, can lifespan of the individuals also be determined by looking at osteocytes? And there's a second part that does bone undergo torsion? And if so, what do osteoclasts aid in the rotation of the bone?
So lifespan of individuals cannot be determined by studying osteocytes, but it can be determined by studying bone histology in general. So if you just cut up a bone of a modern animal or a dinosaur, you can count the rings the same way you would count rings in a tree. And then there are some calculations that you can do to actually figure out which rings have been resorbed by time, and kind of gives you an estimate of how old an animal is. As for undergoing torsion, bone is not great at that. Actually, bone is both stiff and flexible depending on where and which bone we're talking about. Osteoclasts would help in the damage control portion. But what actually would allow any flexibility in bone is its sponginess. So if you've ever chewed on the end of a chicken bone, that end is spongy, and all the trabeculae there actually allow for a little bit of flexion, usually in very particular directions. So I hope that answers that question.
Sounds good. The next question is similar about, I'm thinking it's based on the osteocyte size. Based on bone osteology, can you determine whether T-Rex was mainly a scavenger or a hunter? Guessing based on metabolism?
Probably. So in general, theropods have pretty high metabolism. Some studies show that they're very close to even modern birds. Now, that doesn't actually answer your question, though, because many scavengers have high metabolism too. I mean, and also the scavenger-hunter dichotomy, like the option that it's just one or the other, is not super realistic when you just look at modern ecologies. If you look at certain animals like hyenas and lions, almost all animals will take a free meal, but that doesn't mean that they can't hunt. And that also doesn't mean that their bone biology will reflect that. So a high metabolism animal basically can do a bit of both. And they do have a high metabolism according to literature.
Okay, sounds good. The next question is, do osteocytes vary in size and shape in ganoid-based bone in actinopterygians versus costamine bone based in sarcopterygians? And are there some cells in intermediate taxa which exhibit both types? Right.
So osteocytes tend to be limited to the bone itself, and ganoid, I always mess up that word, is part of the scale itself. Sorry, someone's scrolling this. And so that usually doesn't have a whole lot of embedded osteocytes as far as I know. But the scales have a bony base. So if you're talking about the bony base, yes, the osteocyte shape does vary across. And people don't actually know why. And that's something we're looking into. It might have something to do with scale sensitivity, or maybe something to do with lateral lines. They seem to change shape depending on where in the bone they are. So the closer to the edge, they tend to be sometimes round. If they're in an area that undergoes a lot of stress, they tend to be elongate. So stay tuned would be my answer.
Okay. The next question is, what is your favorite pathology that you have seen, presumably in a fossil, maybe not, and that theirs is a tail pathology in a juvenile Styracosaurus that's currently on display in our museum?
Oh, that's a good one. I think I've seen that one, but I need to go back to the trail soon. My favorite one probably has to be some really weird, like mosasaur pathologies are always really strange. There's a lot of face biting behavior. I love pathologies that also indicate behavior. So things that have tooth stuck in them. So I think there's a hadrosaur vertebrae with a tyrannosaur tooth in it. There's a mosasaur jaw with a shark tooth in it. that kind of stuff is just my favorite because it's like, you don't have to like sit there and scratch your head for too long to figure out what the heck happened, right? Sorry, but so those are kind of some of my favorite pathologies. They're beautiful, they're a really fun story, and it's like direct interaction on an ecological scale. So those are some of my favorites.
Okay. The next question is starts off by saying it's really interesting to see healed fractures in extinct animals. It's easy to imagine how vulnerable an animal would be with a broken bone in the wild. Makes this person wonder what the healing time is for different animals. Do you think a fracture in a frog might heal faster than in a human?
Right. First of all, hi, Kelsey, who also works on similar teeth as I do. But anyway, fracture. So as we know today, frogs and most amphibians actually heal slower than humans, mammals, even some like reptiles and birds. But they have really intense and very interesting immune systems that kind of allow for that to happen. Also, their metabolism can be lowered like on purpose, but also in general, the baseline is lower. So in some ways, they can sequester themselves and heal elsewhere without, having the same pressures of having to go hunt, having to go eat. They have the time afforded to go heal elsewhere without eating and just kind of using their body stores. I think that's why we see so many amphibian pathologies that have survived. Now, I know you didn't quite ask this, but it's also relevant. Like basically pathologies in mammals, in certain mammals, can be indicators sometimes of being social. So sometimes the more social an animal is, the more it can survive really bad pathologies. And this has been shown in modern mammals as well as some paleos. So hope that answers your question.
Okay, great. Another question. I realize a verbal description might not be sufficient here, but this person once worked on an orthomimid phalanx where the distal end had telescoped back into the rest of the shaft. The break was surrounded by what appeared to be swollen, bony callus. Someone else subsequently prepared that callus away, and underneath was smooth bone. What's going on here? Was that callus even bone?
Oh, that's brilliant. I would love to see that. Also, hi, Ian. So basically what happens is, and this actually, if you looked at, I can send you the scan or we can look at the scan some other time, but the fossil frog that I just showed, the underneath is also smooth. And all that means is that it's in a very early stage of healing. So what happens is you have a bone, it breaks, and then a callus will quickly form. And sometimes it's very lightly spongy and easily prepped away, sometimes mistaken for sediment easily. And then only at later times, when the callus is thicker and the bone is in a later stage of healing, will it actually start to pit and take away the old broken bone. And that's when the osteoclasts come in and actually start to remove the old broken bone. So it could have just been in a very early stage of that healing. And so that's why the underneath was still smooth and probably looked like it had just gotten probably the worst toe jam ever. So I think that's what's probably happening with your specimen, but I would love to see it.
Okay, thanks for that. The next question is, what is anosclerotic anosclerocytic bone? I'm not familiar with that term. And what kinds of cells are present? Do you know that term?
Mm-hmm.
Okay.
Yeah. So it's anosclerocytic, and it's basically lacks osteocytes.
Of course it is.
It's fine. Don't worry about it. And so, great question. What kind of cells are present? And why doesn't it have osteocytes, right? So it still has osteoblasts, the bone building cells, and it still has osteoclasts, the bone destroying cells, but it just doesn't have osteocytes, the embedded bone cells. And what's really interesting is that there's a study that just came out a couple of years ago that shows that osteoblasts kind of take up some of the roles of the osteocyte. So what that means is the osteoblast will sit more on the surface and actually sense strain. They'll orchestrate remodeling. They'll do the jobs of the osteocyte. So the genes that were expressed by the osteocyte are now expressed by the osteoblast, and they just got rid of osteocytes being on the inside. Go figure.
Okay, there we go. With my horrible pronunciation. The next question is, do we know if the genes responsible for osteoclasts are pleiotropic? Because that might help answer how they're related to osteoclasts and other cells in their vicinity.
Are genes responsible for osteoclast pleiotropic? Because I don't know, actually. I do believe that osteoclasts have been studied pretty heavily in humans, and I think that is likely true. But I know that in things like sharks, for example, which obviously have lost a bone secondarily, osteoclasts are very different. They're mononucleated, so they only have the one nucleus versus the multinucleated ones that almost the rest of vertebrates have. So I don't know if any of my answers would be like covering all vertebrates. I think in humans that's true, but I really don't know about the rest of vertebrates.
Okay, and there's one final question on our list here, which is, do we also find evidence of fossil instances of hypertumors adjacent to tumors, especially in some of the aggressive malignant sarcomas?
I think the closest example of that would probably be the Terrell specimen. That's some of the most aggressive tumor work that I've ever seen, let alone, so okay, let's start here. Tumors are already very, very rare in the fossil record, probably for several reasons, probably because most of them are not osteoproducing, probably most of them are erosive, so they're just going to show up as holes, and if they're holes, then we probably don't know that they were tumors, because if they were just like a cartilage tumor, and then it disappeared, you just have a hole in a bone, and you're like, I don't know what that is. So there's already a big, big bias there, right? And then if there are other little tumors around it, that would be super interesting. I think the only specimen that I can think of is the Terrell one, where the tumors actually progress downwards into the like shaft of the limb and actually invade the marrow, which probably meant that it had spread through the rest of the body. But that's the only instance I can think of that one. I'll have to think about it, but I don't know of any other specimen that has like a whole lot of extra tumors. That would be super interesting. I always ask when someone gives me like one limb or like one piece, I'm like, where's the rest of the skeleton? Please. Like, I just want an articulated skeleton covered in like at least one continuous pathology. But unfortunately, I have yet to be that lucky. So maybe watch out and hopefully somebody will reach out. But yeah.
Good to know. I'm glad to know the Tyrrell specimen is so unique. Maybe we will see about trying to put that on display at some point so that more people can see it. Okay, thank you for that amazing talk and for handling all those questions. That was great. I just want to, before we sign off, remind everybody that next week, April 13th, we have Dr. Taryn Grant from the University of Sao Paulo in Brazil talking about chemical differences in poison frogs. So thank you very much, Yara, and everybody have a great day.